Our next scholar this afternoon is going to be Deana Moghaddas. Deana is a graduating senior from Del Norte High School. In the fall, she will be attending UCLA and is interested in studying environmental science. In addition to her love for science, she's also a talented musician and has been playing the piano for 14 years. Please welcome Deana.
Good afternoon, everyone. Thank you for attending my presentation. My name is Deana Moghaddas. This summer I had the opportunity to intern at the NOMA Center for Immunobiology and Microbial This summer I had the opportunity to intern at the NOMA Center for Immunobiology and Microbial Pathogenesis under the direction of Dr. Susan Keck and my mentor, Dr. Shixin Ma. And my project was about identifying the interacting proteins of two enzymes known as ACLY and ACSS2. So the Keck Lab studies immunology, which is the study of the immune system, and it's the body's defense against infectious organisms and invaders. Specifically, we study the immune system and its role in aiding and inhibiting cancer. And what we look at specifically within the immune system is the T cells. And these are the cells that are able to frequently interact with the tumor cells. This research supports our knowledge of immunotherapy for cancer, and it allows us to create better chemotherapies. Another side of the lab focuses on immunological memory, and this is the mechanism that allows our body to respond more quickly and efficiently to pathogens that we've encountered before. This research contributes to our knowledge of vaccine development and helps us make more efficient and longer-lasting vaccines. So in our lab, a big focus, obviously, is about the immune responses. We have two immune responses, and one is our innate immunity, and the other is our active immunity. Our innate immunity acts as our passive immunity, so this means it acts as a barrier to foreign particles that try to enter our body. Some examples include our skin and our eyelashes. And once these foreign particles do enter our body counteracts those particles with a general response that aims to limit the spreading of these particles. Some examples include inflammation and stomach acid. Another type of immunity that we have is our adaptive immunity, and this is more specific, and it actively tries to identify antigens, which are proteins on pathogens, and develops a strategic response to these pathogens. This type of immunity takes longer, and it also learns about the threat for future use in order to respond more quickly and efficiently. We have two types of adaptive immunity. One is our humoral response, and this includes B cells that are white blood cells. These circle the bloodstream, identify antigens on pathogens, make antibodies specific to those antigens, and alert the body to fight the pathogen. We also have our cell-mediated response that involves the cells directly, and these include our T cells, which are another type of white blood cell, and these bind to infected cells, either sending messenger molecules known as cytokines to the rest of the body, or by directly killing the cell, which is the process known as apoptosis. So our lab focuses specifically on T cells, and this is because they are the ones that kill tumor cells, but they don't always, which is what we are trying to understand specifically. An influencing factor in a T cell's function is its cell microenvironment, and this is the surrounding environment around the cell. This cell microenvironment can be altered if nutrient consumption is out of equilibrium. So let's say there's a greater amount of fats within the cell microenvironment, then the cell is forced to adapt. And in the process of adapting, tumor cells are able to actually restrict the metabolism of T cells so that they can't uptake the nutrients that they need for their function. So this causes T cells to lose anti-tumor function or their functions in general. What we want to do is regulate the changes to the cell microenvironment in order to boost the T cell's adaptive advantage over that of the tumor cell. So our burning question is, how can we increase a T cell's adaptive advantage over a tumor cell? What we have to look at is a cell's interactions with its microenvironment and also learn more about the mechanisms that occur. So what we looked at are two enzymes known as ACLY and ACSS2. These enzymes are actually in both T cells and tumor cells, but in our project, we are looking at them specifically in T cells, my bad. These enzymes aid pathways for fatty acid and cholesterol production. As you can see, ACLY catalyzes citrate to create acetyl coenzyme, and ACSS2 catalyzes acetate to create acetyl coenzyme. And both of these lead to the development of complex fatty acids and cholesterol. And both of these pathways create the essential sorts of fats for both T cells and tumor cells. But what scientists have found is that in the presence of tumor cells, the expression of these enzymes are reduced, which is what causes the T cells to have a decreased metabolism in the presence of tumor cells. Because these pathways are crucial for their metabolic function, T cell effectiveness is then decreased. Since our tumor environment has reduced expression of ACLY and ACSS2, we want to boost the function of these enzymes. So as you can see before, we have reduced expression of ACLY and ACSS2 when the T cells are in the presence of the tumor cell. And what we want to do is boost the function of these enzymes. And hopefully what we are hoping for is that if we boost the expression of these enzymes, that we boost T cell function over that of the function of the tumor cells. And if we want to boost the enzymes in these pathways, we first have to know a lot more about them. So what we're looking at is identifying the proteins that interact with our enzymes. So our enzymes are proteins, but we're looking at even the smaller proteins that aid these enzymes. Identifying the specific interactions that occur can give us a lot more knowledge in how we can intercept these pathways and possibly boost the expression of these enzymes as well. So how we went about this was an experimental design that included first plasmid preparation, then transfection, immunoprecipitation, a Western blot, and mass spectrometry. We first begin with plasmid preparation. We do this to create a construct that contains our genes of interest, ACLY and ACSS2. We used a plasmid vector, which is a circular DNA molecule used as a vehicle to carry foreign genetic material into another cell. So this plasmid vector includes bacteria so that when we infect the cells that we're using, that our genes can produce the proteins that we want. So we did this by starting with our original plasmid, cutting out the irrelevant DNA that we didn't want to use with restriction enzymes, then inserting our genes of interest within these vectors, and then we ended up with 3 groups of vectors. One is our control group, used as a base comparison for our other groups, a group with our gene of interest ACLY, and one with ACSS2. Before we can move on, however, we wanted to check that we had indeed made the plasmids that we wanted. And to do this, we sequenced our plasmids. We wanted to check that we had made the correct vectors, so what we did was correspond the code for our vector DNA with that of our known ACLY and ACSS2 code. And what we can see is that the nucleotide sequences are the same. So DNA is double-stranded, and we just use the forward strand in this picture. But as you can see, these are the same code, and same with ACLY right here. Our next step was transfection, and this was infecting our human cells with the vectors that contain our bacteria and genes of interest to express our protein of interest for further testing. So as the cells would uptake this vector, they would use their own machinery to actually express the protein that we want to identify. Before we can move on with this step as well, we had to check the transfection efficiency. So in the plasmid preparation, we added a fluorescent known as GFP in the preparation step, and so that the infected cells would exhibit a bright green color. We then observed the cells to see that approximately 70% of our cells had been infected via the bright green color. And if so, and luckily by our observation, they were properly infected, and then we were able to move on to the next step because enough of our protein had been expressed. Our next step was immunoprecipitation. So our cells have thousands of proteins, and what we're trying to do is isolate 2 specific proteins, ACLY and ACSS2. First, we used a cell lysis to empty out all the contents of the cell. And then we added antibodies that are specific to ACLY and ACSS2 that would bind with our proteins of interest. At that point, we added magnetic beads that would bind to the antibodies, and then we used a centrifuge to spin these test tubes down and collect a protein antibody bead mixture that ended up at the bottom. We then removed the beads using a process called buffer elution, and then we were able to isolate the proteins that way. So at that point, we had ACLY and ACSS2 isolated. And then again, to move on to the next step, we had to see that we had collected enough proteins to identify even the smaller proteins that interact with ACLY and ACSS2. And we did this using a Western blot analysis to validate the precipitation of our proteins. So what we did was add our proteins into a gel and then transfer that to a construct that includes a sponge, a membrane, and filter paper. At that point, we stained our construct with a primary and secondary antibody specific to our proteins, and that would allow for the detection of our proteins when imaging. And this is our ACLY Western blot. So what we want is for our immunoprecipitated lane to show a greater concentration of protein than our whole cell lysis lane. And over here, you can tell that the immunoprecipitated lane is a lot dimmer than that of the whole cell lysis. So for the sake of time, we actually threw out this protein sample because there had been an error. And what we concluded was that the antibody that we were using for our Western blot wasn't up to par with the quality that we wanted. And so for the sake of time, we threw this one out. But for our ACSS2 sample, we could see that the immunoprecipitated lane was a lot more concentrated than that of the whole cell lysis lane. So that means we did overexpress our protein enough for the next step to identify the even smaller proteins that interact with ACSS2. Our last step was mass spectrometry, and this is using a magnet to separate different molecules and measure their masses in our sample of ACSS2. We then correspond the mass to ratio charge of these molecules with the known mass to protein charges of proteins that we know to identify what these specific proteins are. So our results, we actually won't know for another 2 to 3 months. I know I took you guys all through that, but yeah. But here's a sample graph. So as you can see, the peaks is what we're going to be looking at. And for this, the mass to charge ratio is 29. So we would correspond that with the mass to charge ratio that we already know. So our next steps, once we get our results, we want to see what role do these interacting proteins play when actually interacting with the enzyme. So at this point, we would know what these proteins are, but now we have to see what they do in context. So what do they actually do when associating with our enzyme? And we also want to see, can we possibly use these interacting proteins to regulate T cell function? So we are hoping to see that maybe we could use these interacting proteins in a way to boost our enzyme function and then maybe boost our T cell function. So before I finish, I would like to extend a great thank you to everyone that attended, as well as an even bigger thank you to my mentor, Dr. Shishing Ma, Dr. Susan Keck and the Keck Lab, our advisors, Donna Mapston and Madison Dodds, all the Salk scholars who made my experience so amazing, the Life Science Summer Institute, the Heithoff Family Foundation, Mark Kragnim and AccuCenter, and also, I didn't put this up there, but my family, who, yeah. Okay, thank you so much for listening.